Every living thing is built from DNA, the remarkable molecule that serves as the blueprint for all life on Earth. DNA is a set of instructions that shapes life, determining everything from the color of your eyes to how your body fights disease. For decades, scientists could read DNA, unlocking the secrets hidden in our genes, but they couldn't easily change it. It was like having a book full of typos and mistakes, but with no way to correct them, no matter how obvious the errors were. Enter CRISPR, a revolutionary tool first discovered in bacteria, where it acts as a natural immune system, protecting them from viral invaders. Scientists realized they could harness this bacterial defense system to edit the code of life itself, making precise changes to DNA in plants, animals, and even humans. CRISPR isn't just for scientists in labs, it's a technology that could touch all our lives. Imagine a world where we can cure genetic diseases, help families avoid inherited illnesses, and grow crops that withstand drought and pests. Or picture creating new biofuels to power our future, reducing our reliance on fossil fuels. CRISPR makes these possibilities real, not just science fiction. By understanding CRISPR, you can join the global conversation about how this technology will shape our future, from medicine to agriculture and beyond. The story of CRISPR began with a simple curiosity about strange, repeating DNA patterns in bacteria, an observation that seemed unimportant at first. That curiosity sparked years of research, leading to the discovery of one of the most powerful tools in modern biology. Now anyone can grasp the basics of how CRISPR finds, cuts, and rewrites genes, making genetic editing more accessible than ever before. You don't need a PhD or a lab coat to appreciate how this tool works. Just a willingness to learn and a sense of wonder about the living world. CRISPR empowers us to see life at the molecular level, revealing the intricate machinery inside every cell, and giving us the ability to understand and even rewrite our own biology. Let's break down how this remarkable tool works, step by step, and explore the science that's changing our world. The journey from a bacterial defense system to a world-changing technology is a testament to the power of scientific curiosity and human ingenuity. And now, as we stand at the frontier of genetic science, we're all part of CRISPR's unfolding story, one that could shape the future for generations to come. To truly understand how CRISPR works, let's meet its three main players: Cas9, guide RNA, and the PAM sequence. Each of these components has a unique and essential role, and together they form the core machinery that makes gene editing possible. Cas9 is often called the molecular scissors. It's a specialized protein that can cut DNA, but it doesn't just cut anywhere. It only acts where it's specifically directed. Without guidance, Cas9 would be like a pair of scissors floating in a library, not knowing which book to open or where to snip. That's where the guide RNA or gRNA comes in. Think of it as the GPS navigation system for Cas9. The gRNA carries a code that matches a specific DNA sequence, leading Cas9 to the exact spot in the vast genome where a change is needed. It's like entering precise coordinates into a GPS to reach a single house in a sprawling city. Without the guide RNA, Cas9 would be lost, unable to find its target among billions of DNA letters. The gRNA is what gives Cas9 its remarkable precision, ensuring it only acts where we want it to. Scientists design the gRNA to match the exact DNA sequence they want to edit. This customization is what makes CRISPR so versatile. It can be programmed for almost any gene in any organism. But there's an important checkpoint: the PAM sequence. This is a short stretch of DNA, often just 3 letters like NGG, that must be present right next to the target site. The PAM acts as a signal telling Cas9, you're in the right place. Cas9 won't cut unless it finds this PAM sequence immediately adjacent to the target. It's a built-in safety feature, preventing Cas9 from making random or accidental cuts elsewhere in the genome. You can think of the PAM as a security checkpoint, making sure Cas9 only acts in the right place at the right time. This extra layer of control is crucial for the accuracy of gene editing. The system works because of this 3-part harmony: Cas9 cuts, gRNA guides, and PAM confirms. Each part is essential, and together, they create a powerful and reliable editing tool. This incredible precision allows scientists to target a single gene among billions of DNA letters, making changes with a level of control that was once unimaginable. The PAM sequence acts as a crucial safety feature, preventing off-target cuts and reducing the risk of unintended genetic changes. It's one of the reasons CRISPR is considered so safe and reliable. When these components work together, CRISPR becomes a powerful, accurate gene editing tool that is revolutionizing biology, medicine, and agriculture. With this trio, Cas9, guide RNA, and PAM, we can now rewrite the blueprint of life with a level of precision and control that was never before possible. Understanding these three players is the first step to grasping the true power and potential of CRISPR technology. Now let's explore how these components work together in real time to find and edit the right spot in the genome, unlocking new possibilities for science and medicine. Once inside the cell, the Cas9 gRNA complex begins its search. This search is not random. It's a highly organized and efficient process honed by evolution and refined in the lab. The Cas9 protein, guided by its RNA partner, moves purposefully through the dense environment of the cell nucleus, where countless DNA strands are tightly packed together. The nucleus is crowded with billions of DNA letters, but Cas9 doesn't read them all. Instead, it uses a shortcut to quickly narrow down its search, saving time and energy. Instead, it scans for the PAM sequence, the first sign it's in the right place. The PAM, or protospacer adjacent motif, is a short DNA sequence that acts like a beacon, signaling Cas9 to slow down and take a closer look. When Cas9 finds a PAM, it pauses and checks the DNA next to it. This pause is crucial, as it prevents Cas9 from making mistakes and ensures it only investigates promising locations. The DNA unwinds, and the gRNA tries to match its sequence to the target. This is a delicate process, as the gRNA must align perfectly with the DNA for the next step to happen. If the match is perfect, the complex locks in; if not, it moves on. This ensures that only the exact genetic address is selected, avoiding accidental edits elsewhere in the genome. This two-step check, PAM first, then sequence match, ensures accuracy. It's like a security system with two locks, both of which must be opened before entry is allowed. Only when both checks are passed does Cas9 prepare to cut. This careful preparation is what makes the system so reliable and trusted in genetic engineering. This process prevents dangerous mistakes and off-target edits. By double-checking, Cas9 protects the cell from unintended consequences, making gene editing safer. The specificity of this search is what makes CRISPR revolutionary. It allows scientists to edit genes with a level of precision that was once unimaginable. It's a molecular treasure hunt, with safety checks at every step. Each stage is designed to confirm the target and prevent errors, ensuring only the right gene is edited. The system only acts when it's certain it's found the right address. This confidence is what gives researchers the power to make precise changes in DNA. Now, with the target confirmed, it's time for the moment of truth. Cas9 is ready to make its precise cut, setting the stage for the next step in gene editing. With the target found, Cas9 shifts shape and activates its cutting blades. It snips both DNA strands, creating a double-strand break, CRISPR's central event. This cut is precise, happening just upstream of the PAM sequence. The break signals the cell's repair machinery to rush in. Without this cut, nothing changes. With it, gene editing becomes possible. The cell must now fix the break, opening a window for scientists to guide the repair. Cas9's job is done after the cut, but the real editing happens next. The cell's emergency response determines what happens to the gene. This deliberate break is the key to rewriting DNA. The next step, how the cell repairs the damage. The cell treats a double-strand break like a major emergency. Specialized proteins rush in, protecting and preparing the broken DNA ends. Some hold the DNA together, others clean up the site, and some stitch the strands back. CRISPR hijacks this natural repair process, pointing the cell to a specific spot. The cell doesn't know the break was intentional, it just fixes what's broken. The repair pathway the cell chooses determines the outcome of the edit. There are two main options: a quick patch or a precise replacement. The first repair option is non-homologous end joining, or NHEJ, a quick patch job. NHEJ simply glues the DNA ends together, often introducing small errors called indels. These errors can disrupt a gene, effectively turning it off. Scientists use NHEJ to knock out faulty genes, stopping harmful proteins. The second option is homology-directed repair, or HDR, a precise fix. HDR uses a template to repair the break perfectly, letter by letter. Scientists can supply a custom template to introduce specific changes or corrections. HDR allows true gene editing, rewriting the code, not just breaking it. For diseases caused by a single DNA typo, HDR can correct the mistake. The cell's choice between NHEJ and HDR shapes the final genetic outcome. NHEJ is fast but messy, HDR is slow but precise. Both pathways are essential tools in the CRISPR toolbox. Let's see how HDR can cure a real genetic disease. Take sickle cell anemia, a disease caused by a single DNA typo. One wrong letter in the hemoglobin gene deforms red blood cells, causing pain and damage. CRISPR offers a way to fix the root cause, not just the symptoms. Scientists collect stem cells from the patient and treat them with CRISPR-Cas9. The guide RNA leads Cas9 to the faulty gene, where it makes a precise cut. A repair template with the correct DNA sequence is provided. The cell's HDR machinery uses this template to fix the typo, restoring the healthy gene. Edited stem cells are returned to the patient, producing normal red blood cells. Clinical trials show patients can become free of painful crises. This is real-world gene editing, CRISPR rewriting biology for the better. It's a glimpse of what's possible for many genetic diseases. Cas9 started the revolution, but the CRISPR toolkit keeps growing. New proteins like Cas12, Cas13, and Cas3 offer unique features. Cas12 makes staggered cuts and recognizes different PAMs, expanding editing options. Cas12's shredding ability is used for sensitive diagnostics, detecting viruses or mutations. Cas13 targets RNA, letting us temporarily silence genes without changing DNA. It's like a dimmer switch for gene activity. Cas3 acts as a DNA shredder, deleting large sections, useful for removing stubborn viral DNA. Each new tool brings more precision and flexibility to gene editing. The CRISPR toolkit is evolving, opening new frontiers in science and medicine. The possibilities are expanding faster than ever. CRISPR has transformed our ability to shape the living world. In agriculture, it's creating hardier, more nutritious crops. In environmental science, it's helping clean up pollution and produce sustainable fuels. But with this power come ethical questions about safety, access, and the limits of editing life. Germline editing, which affects future generations, is especially controversial. Most scientists agree. We need public debate and careful oversight. Meanwhile, CRISPR research is advancing rapidly, becoming safer, more precise, and more accessible. New therapies for diseases like muscular dystrophy and blindness are on the horizon. We're witnessing a turning point in science and medicine. Understanding CRISPR's step-by-step process helps us make informed choices about its future. This isn't just a story about technology, it's about human ingenuity and the power hidden in our DNA. The future of gene editing is already here, and we all have a role in shaping what comes next.